Okay, hi everybody. I'm Tyler Morgan. I'm a staff scientist in fMRIF, which is the fMRI facility. And along with Renzo Huber, today we're going to be talking about high resolution MRI and fMRI. So we're going to start off with a little bit more of the technical side, leading out of what we heard on Tuesday about physics from benign. And then Renzo is going to hop into what we can do using high resolution in functional imaging using MRI. So just to start off, wherever my mouse is, perfect. I'm going to ask a really simple question, which is just how can we actually increase resolution in MRI? So I think everybody's gotten the idea so far, but it's really important to remember throughout every experiment that you ever do that MRI is a technology of compromise. You can never get everything. It's impossible, but you can choose what's important for the applications that you're actually looking at. And so just to give an example of this, there are kind of two main ways at the basics of MRI that you can increase your resolution. The first is you can scan longer, and that's kind of demonstrated here. This is a really, really beautiful data set from Magdeburg where they scanned one single person. It's not one single scan, but the way they did it is kind of tricky, but they got 250 microns, so 0.25 millimeters resolution. It's one of the highest in vivo scans that's ever been done in a human scanner, but it took 3.5 hours. And that's a long time for people to stay in the scanner to get one scan out. Alternatively, you can also increase your field strength. And this we went over a little bit in Vinay's lecture on Tuesday, but essentially you get more signal in less time if you go to higher field strengths. And I know a lot of people probably have a little bit of something in mind when they think of higher fields in MRI, but just to give you an overview of kind of what has been done and what has been tried, we can kind of see the full gamut of everything above 3T here. People have tried 4T scanners. Technically, I skipped 4.7, but there's also 5, 7, 9.4, 10.5, 11.7, and soon to be efforts towards 14. And I'm concentrating specifically on human scanners here. And this is kind of a cool thing. Each of these field strengths has their own really, really good things about them. Each of them has things that are really hard to overcome. But just to give an idea of what's really important in this building, we have 7T high field scanners. We also have an 11.7 Tesla high field scanner. And just to give you an idea of the accessibility that goes along with these two things, the 11.7 is one of 3 systems in the world that exists at this field strength for humans. At the same time, in that same corridor, there are 3 7Ts. There are also 4 accessible 3Ts in that corridor as well. So like you kind of have these like really far ends where the technical challenges are extreme, but they have some really, really great payoffs if you can get there. And at the same time, we also have some scanners that are basically compromises. They really have great things about their signal amplitudes and what you can do on them, but you can also like build them and engineer them and manufacture them, etc. So just to give you an idea of where we sit. But I'm going to come back to that original question, which I think is an important one, which is if you only have these 2 dimensions that you can work with, are there any shortcuts that we can take in order to actually try to skip part of this? Not go to higher field strength to go to higher resolution, because that's not very common in our field. And at the same time, not just scan longer and longer and longer. Like if you wanted to look at ex vivo studies, do you have to study for 7 days or whatever? No, oftentimes we will do something in between. And so just to kind of walk you through this and give you an idea of how people came to trying to figure out how to speed up and go to higher resolution at the same time, the basic thing that we want to do in MRI, which we saw again a lot on Tuesday, is that we want to record from the frequency space, which is K space in our particular circumstance, And we use this to reconstruct images that are of whatever body part that we want to do. In fMRI, this is most frequently the brain. And we also heard, so just I'm posing a question up at the top, that doesn't give us any extra speed. Like we know nothing about how to speed this up until we get to the point which we saw on Tuesday again, that it's actually pretty cool that you can just skip lines in MRI. And this is one of our main abilities to actually go faster. So if we want to go to higher resolutions, we can just kind of like skip every other line. And then you've kind of extended your scan time because of the resolution, but you've shortened it in half because you cut every second line out. However, this comes with a pretty well known for anybody that's run scans, but annoying artifact, which is that because we're recording in frequency space, anything that is outside of the field of view, and those skipping lines are modifying the field of view, is going to be in the form of a fold-over artifact. So you can see here that where A and B are labeled on the left and right-hand side, they're switched here because basically the right-hand side of the screen is coming through the left and vice versa. And this makes it kind of hard to do a brain imaging study on this particular field of view. So this is maybe a good, maybe bad choice of words, but in parallel to the idea of going faster in MRI, we've also for a long time been working on engineering projects called parallel imaging. Parallel imaging is the idea that we can actually read out spatially from different places in an MRI coil in order to increase our signal to noise. So just to give you an example of this, this is kind of a toy example, although coils like this have existed in the past. You can have smaller coils than just covering the entire head. And in this particular case, if you have one on the left-hand side, I guess it would be right-hand side and left-hand side of the person's head, those coils are actually more sensitive to the side that you're showing them on, or that they're actually closer to. So this example here is basically saying that if we reconstruct this image from each of the coils separately, we would get an image that is very bright on one of the sides in one of the coils, and the other side on the other coil. And then normally what we do is we just combine those things. I'm showing the toy example with two just because it's easier to think about a little bit, but of course, in real life, people have extended way beyond this. There are a lot of different options downstairs in the NMR center, but here I'm showing an example of the 3D model of a 32 channel head coil. So you kind of have very specific spots on the surface of the brain that you're sensitive to with each of those coils. Okay. So we've gone over kind of two different things here. We've gone over the fact that there are parallel coils that we can use in order to increase our signal. And we've also gone over the fact that when we try to accelerate by skipping parts of K-space, we're actually just creating fold-over effects. Now we're going to combine those two things into how would we actually solve this problem. So the acceleration problem in MRI is essentially just that if each of the coils is recording those folded over versions of the brain, those are going to be different for every single coil that you've recorded from. And really what we would like to do is take that information and kind of separate it so that we can reconstruct something with less time than it would normally take to fill that entire K-space. And we can do this through accelerating using the coils. So just to review that, how do we go from the fact that these are the data that we're actually recording to actually solving for this regular image, which is the brain that's actually inside the scanner? There are a whole big slew of different types of algorithms that do this. I'm going to cover kind of 2/3. And they're ones that have been around for a very long time. They're also the ones that you are most likely to encounter on the scanner. I actually don't know anybody who gets away with not using some form of acceleration for most of people's studies. Okay, so first up, we have what's called sense or asset, depending on what type of scanner you're on. Sense slash asset, I'm just going to call it sense, is based on the fact that you can actually calculate what the sensitivity maps of each of those coils that you're using to measure looks like in 3D space or 2D space or whatever it is that you're measuring it. So here on the left-hand side, you can see that I have labeled coil sensitivity maps. This is essentially just where in the space that the person's head is in are you most sensitive to with that particular coil. So the white space is that the coil on this side can see mostly that side, and the opposite here. Now, interestingly, the way that the sense algorithm is set up, you want to create a system of equations, and I purposely avoided putting an actual formula in here, but you can kind of just get the picture with what the voxels look like. We're recording these green voxels. So if we take one single voxel in all of the channels that we've recorded in the data that we got, which are these folded over versions, we can create a system of equations that relates what that would look like in the full image to what it looks like in the measured images, using the fact that we know something about the spatial layout of where those are in space. Just to review that, basically what we're doing is we're saying that the green voxels here, those are the equivalent of if this blue voxel here was multiplied by whatever is actually in that voxel in real life, plus whatever this red voxel in the sensitivity map is, multiplied by the purple one. And the key here, obviously, because if you only had one of these lines, you couldn't solve that. The key here is that if you have, let's say, 32 of these equations, you can actually solve for a pretty high acceleration. Does that make sense to everybody? I'll stop for questions in a little bit, but feel free to jump in if you have anything. So this idea is essentially just that you can accelerate as much as you have coils. So you have many, many coils in your receiver. You can actually accelerate close to that. There are some limits and real world problems, but you know. Okay, the next one that we're going to go over is grapha. And I suspect a lot of people have heard of this, or IPAT is also what it's sometimes called, although IPAT is a little bit more general. Grapha and sense are very, very similar in what they're trying to accomplish, but they do it in dramatically different ways, primarily just because grapha concentrates on trying to fill in our data, not in the image space, not in the brain space that we're looking at, but in the case space directly. So it tries to skip over having to do a reconstruction prior to solving the problems of getting the voxel values. And so what we're going to do here is you can see, just in this little toy example again, let's say we're recording all of these red voxels in case space, and we're skipping lines here. But every once in a while, and specifically in the middle of the image, we will often collect a kind of series of lines that are continuous. So we're not going to skip any lines in the middle. This helps us save some of the time by skipping lines elsewhere, but considering the middle of case space as special. And it's also important because this is where we're going to train a model that is going to try to fill it in. So in the sense example, we were trying to set out a set of equations that we're trying to take voxel values that we knew or recorded and trying to untangle them. Here, we're doing something similar. So we're taking basically all of the different coil elements, and we have the same kind of case space in those coil elements, and we're going to use all the other coil elements to try to train a model to fill in the case space points that are missing. And you can kind of see this here. So let's say each of these little squares is a zoomed in version of what we're seeing here. And we're going to use all the points that we have in order to try to fill in the point that we're missing. And we'll do this for every point that is missing. Okay. So we didn't record these data, so we know that there are going to be some limitations, because as in every other part of science, anytime we try to estimate things, we don't always get it right. The first important thing to always remember is that your imaging techniques, and specifically parallel imaging techniques, are always limited by the number of receivers that are in your coil. So that's an important factor to know when you're planning studies. So basically, if you're running on, let's say, a birdcage coil and it only has 6 or 8 elements, you cannot accelerate more than that. You can't solve that problem. Alternatively, if you're on the best, coolest coil in the entire world and you have 128 channels, you can do a lot more acceleration because you have a lot more data that you're technically playing with. It often doesn't look like this from the user's point of view, because basically what we get out is the reconstructed total image. But internally, you have 6, well, let's say 32 to 128. That's 4 times as much data that you're actually playing with to solve these models. The second thing to always remember is that noise is cumulative. And what I mean by this is that no model is perfect. So the more you accelerate, the more points you have to try to fill in, the bigger chance that you will get some of those points wrong. And so basically, we can actually map these things out. This is called a G factor penalty. G in MRI almost always stands for acceleration of some kind. But essentially what this is telling us is that we saw all of those sensitivity maps. There are some areas of the brain where those sensitivity maps basically all have similar values. If you're, let's say you have a coil right on the top of the parietal on one side of your head, and you're trying to solve for a voxel that's right there, it's pretty easy because you know which coil is the best coil for it. But if you're doing something in the brainstem, let's say, all your coils are roughly the same distance from it, and they probably have the same power in explaining those voxels. And in those areas, if you accelerate a lot, you tend to be making worse predictions more often. And so this is something we have to deal with. The last thing that I'm going to say as a limitation, even though this list could probably be longer, is that algorithms are never perfect. That's something to emphasize. Some wrapping artifacts will almost always be present when you're solving these types of problems. And just as an example of what that generally looks like, I'm going to move on to another type of filling in algorithm that is using a slightly different principle, but we're going to start with just pointing out that, like we saw on Tuesday, and also you saw it in a previous slide, if you're skipping lines like you are here, your artifacts are always going to fold over in the exact same position. They are what's called coherent. So in other words, you are actually forming an image with the data that are wrong in your model. Sometimes it is useful to try to figure out how you can actually make that kind of artifact that is coming into the field of view completely incoherent. And that's what I'm sure a lot of people have heard about this as well, but compressed sensing tries to do. Compressed sensing is the idea that we can rearrange the points that we are looking at in k-space so that every time you're trying to fill in a position, it is kind of randomly oriented in comparison to the points that you have. And that way, whenever it's folding in, each point will fold in a different direction and a different magnitude, which is kind of a cool idea. And you can kind of see an example of that here, which is essentially just that rather than having a Cartesian grid, you're basically just choosing points as randomly as you possibly can, because then your foldover artifacts are still there. They're identical. As long as the number of points here and here are the same, the magnitude of foldover artifact is essentially the same, with some exceptions. But what you can also see here is that you can much better see which brain is the real brain here. And the reason is that all of your holdover artifacts are not forming a brain properly. So the only one that you're left with was the one that was in the original signal. Does that make sense? Okay. And this is kind of cool because it allows you to push further, excuse me, your acceleration without as much loss as you would typically see because you're trying to overcome artifacts. And I'll just give an example of this from some data that we collected very recently. And that's that on 7T-B, which is our Terra 7T, we have a compressed sensing algorithm for doing anatomical imaging at 7T. That was kind of repetitive. But the point being, if we take as our example for comparison the GRAPPA3 reconstruction, so this is, in other words, skipping 2 lines for every line that we've collected, we can do this scan in about 9 minutes and 37 seconds for an anatomical that has sub-millimeter resolution. So this is 0.75 whole brain, also MP2 range, etc. Using compressed sensing, we can cut a lot more time out. And at the same time, that always equates to more case space out. You can see here that I've calculated it. The GRAPPA3 kernel is using about 35% of the original case space and trying to fill in the rest of it. The compressed sensing one is only filling in 18%, but it can overcome a little bit more. And so I'll let you judge for yourself. I think that those look very similar personally, and it saves you 3.5 minutes at the scanner. It also means that you're less susceptible to things like motion because just you're not scanning for as long. The chances of someone moving are less. So I'm going to wrap up here and essentially just say that once we have accomplished this in the anatomical world, we can start moving on to what a lot of us care about in this course, which is that we want to be able to measure brain function. And for high resolution, that oftentimes means we want to get to layers. So here I'm showing examples of what you can do if you have higher resolution scans. And that's essentially that you can label your tissues very well. You can get an estimate of where gray matter is and is not very well. And then you can calculate things like layers or the size of columns. You can do all kinds of stuff with the metrics of what the cortex looks like. And here are some nice 3D models as well. And with that, we're going to move on to Renzo talking about all the exciting things that you can do in functional. Jason.
Why 32 coils? I mean, why doesn't everyone just use 120?
A couple different reasons. I mean, there are people who do explicit coil building here, and they'll be able to answer that better. One of the biggest things is that you're inside a giant magnet. There's not a lot of room. Ideally, we would probably have the largest phased array that we could possibly have, something like what areas of physics like astrophysics use. They like try to increase the number of elements as much as they possibly can. We are always working under the limitation that we have to build something that fits, 1, around the person's head, and 2, is not bigger than the space that we have inside the MRI. So you can build bigger and sometimes better coils if you have larger cores. But obviously at high field, this becomes a challenge again, because then you're dealing with other things that are really problematic, like D1 becomes really, really difficult to handle if you're trying to go to high field and you have a large core. So it's just a balance.
Is there any significant penalty because we save all of this?
Yeah, so that's what we were talking about a little bit with the G factor penalty. So you essentially lose, it's not necessarily that you lose signal, but you have increased contributions from noise that hurt your signal.
From online, someone else does CS suffer its own artifacts. I saw that it has gives rate.
It does, yeah, absolutely. There are different forms of compressed sensing. The one that we have is technically a Cartesian readout that is using compressed sensing. And that one in particular doesn't have a lot of ability to fight big motion if it happens during the scan. the main way that we're trying to compete with things like raining from motion artifacts is to shorten the scan.
This Chris also wrote up for the future poster to
Yeah. Sorry, so they didn't hear me at all. Sorry about that. Yes. So basically, there are different implementations of how compressed sensing works, the different acquisitions that are matched with it. Our particular one is no more or less sensitive to motion than, let's say, the grappa example that we showed alongside it. The only big difference is that we've shortened the scan time, and that allows us to combine motion things. Also, I will say that if you use compressed sensing algorithms and you really push undersampling, your images will have artifacts. There's no real way around that. But they look a little bit different, and they're more based on the types of fitting that you're doing. They're not based on the same artifacts that we were talking about here. But I'm happy to talk about that more offline.
Hello. My cursor is here. Can you see that? Great. So I'm working with Kyla. We're in the group of Peter Bandettini, and I will specifically focus on high resolution with respect to functional imaging. And functional imaging is necessary across different spatial scale because the brain works across different spatial scales. So we know that the brain has all these different areas that do different things. And as exciting as it is to kind of look at these brain areas and what they do and what we learned already with fMRI about mapping these brain areas, sometimes it's frustrating how little we learn about the brain by just looking at these blobs. And some of you know this metaphor, but some people say that trying to understand how the brain works by looking at these blobs is somewhat like trying to understand how a truck engine works by looking at it with an infrared camera. You know kind of which brain areas are consuming energy, like you see that when the truck is driving, the front is heating up, which is cool, but it doesn't really tell you how the truck engine works, right? We need a different spatial scale of the densely packed microcircuitry to get a better mechanistic understanding of the brain, which is why we need high resolution. So we need more than just the voxels across the different tissue types, gray matter, white matter, CSF, but also we need activity distributions within the gray matter across these different cortical layers. Because these different layers, they contain different neurons, and those different neuron populations have different processings. So going from conventional fMRI at these resolutions to layer fMRI resolutions, we can subsample these different neural populations, which is giving us important new information to understanding how the brain works, namely in the sense that we not only learn if a given brain area is involved in a certain task, but we learn something about where this activity is coming from, how it's following the different processing step, how it's modulated by input from other areas until we have a new output of this computation with respect to feedback and so on. So going from conventional resolutions to high resolution not only tells us which areas are connected to each other and which of those connections are stronger than others, but importantly, it tells us something about the causality, the directionality of these connections, which is useful information to have. Now, to get to these resolutions, however, we need to rely on a lot of methodological advancements over the last decades. And I would like to exemplify this by looking at astronomy. So we have this beautiful night sky over there. And if you have a very dark spot somewhere in the nature out there, you can see that some parts of the sky are just brighter than others, right? And then you can zoom in and see that there are these beautiful clouds. And you zoom in further, and you see there are subclouds, and so on and so forth, until you ultimately hit the resolution limit of earthbound telescopes like La Silla in Chile here. Now, with decades of methodological improvements going to space, you can see that there's much more information to obtain. Namely, you see that it's not just a cloud, but there are all these wiggles along the contours over there. And then not only going to space, but then also tweaking the methodology and software quite a bit, you can see that there's even more information a decade later visible. Namely, not only at the contour you have these wiggles, but you get a better understanding of the dynamics of these clouds. These wiggles are everywhere. And I'm showing you this example because this is just about the time scale of fMRI methods developments. Last week, we heard from Peter Bandettini about the first experiments in humans. which was a huge leap forward, and they could non-invasively capture these brain systems, right? Which is awesome. And then with decades of improvement, for example, with a lot of hardware improvements, people achieved resolutions like 2 orders of magnitude better. And then still with very similar hardware, with a lot of software improvement sequences, still there's another order of magnitude in the voxel volume reduction. And there's this Moore's law of fMRI resolution that every decade or so you get about 1 order of magnitude reductions. And in these spatial scales, you have additional features. And of course, each different spatial scale has its unique window to investigate brain function. Like here we're looking at brain systems, right? Not even separating sensory and motor cortex, looking at brain systems, which has taught us a lot about how the brain works in the last decades. Going to these 1 millimeter resolutions, you still kind of are working on in a simplified neural model. You still need to assume that all the neurons across the entire cortical depth are kind of doing the same thing, but you can resolve it maybe across the topology and not worry about kissing chirae, which is already better than assuming that all the neurons in the entire brain area are doing the same thing, right? And at these layer resolutions, still you're working with kind of simplified models, but you don't need to assume anymore that all the neurons across the cortical depth are doing the same thing. But still, even with those voxels, you have more than 1,500 neurons per voxel, and you cannot be so naive to think that all of those 1,500 neurons are doing the same thing. But we are better off than the other resolutions. And specifically at these resolutions, we are aiming for features like these stripes over here, the layers, but also these kind of columnar features or vascular features. And we can try to interpret them with respect to kind of neurons firing. We can try to interpret them with respect to the kind of the arteries being dilating or the veins straining, the oxygenation and everything. And in the lecture end of this month, we will specifically talk about how to interpret these kind of signals with respect to contrast mechanisms and how to analyze those signals. In this lecture, I want to rather focus on the high resolution part and give you an idea of why we want to go there. What's the new information that we get from these high resolutions? And I would like to show you this with this experiment here, looking at the motor cortex. So at conventional resolutions, when you do a finger tapping experiment, you see that we have this blob of activity in the sensory motor system. Now, if you ask a person to just think about moving their hand without actually moving the hand, making it more kind of neuroscience-y, you can see that the brain pattern actually looks frustratingly similar, right? It's a bit weaker and skewed differently, but how can the brain look so similar when the behavior is completely different, right? So to solve that, we can zoom into this tiny patch of the cortex and increase the resolution further and further and further until we have these different stripes, the layers. And we know from tracer studies, specifically in animals, that the superficial layers over there are the ones that receive a lot of cortical input, like sensory feedback, motor planning, and so on. Whereas the deeper layers are the ones with the giant bed cells that go to the spinal cord and trigger the muscles to move. Seeing both here for this task. Now, when we look at these imaginary tapping, we don't really have this movement, right? There's no motor output. So you can actually see that now with the higher resolution information, we not only know that both times the same area is kind of involved in the task, but now we understand how it's differently involved. We get a better understanding of kind of what's happening under the hood with respect to the truck metaphor. And you can use these kind of models of kind of different layer groups doing different things for all kind of different tasks. Here, for example, looking at tapping versus touching, where in one case, in this tiny patch of the cortex, we have kind of 2 layers. In the other one, we have only 1 layer, and the other one is actually kind of inhibited already. The reason why I'm showing you this example here now is to make you aware that there's an entire additional information aside of the layers that high resolution can give you, namely the columns. So while this kind of patch has this nice dissociation between upper and deeper layers differently activated for the different tasks, if you move just a few millimeters further towards the left, you can see that this nice dissociation breaks apart, right? So there is not only the layers, but there's also the columnar units that might include a lot of additional information. I want to show you one example of how high resolution might be useful there with respect to the toy model of the motor cortex again, showing you this kind of animation here where we are looking at a tilted axial-like slice where we have the central sulcus. And on the left side, the thing that you'll see growing out is actually a flooding algorithm on the sensory cortex. And on the right side, we have the motor cortex also kind of flooding to look at this now in a topographical space to not only look across depth, but basically the surfaces in voxel space though. And then we can rotate this around and kind of look at this now from the perspective of the back of the brain. So this would be the midline of the brain, and here we would have the right ear. And then I can project the individual fingers onto it. Now on the sensory cortex, the pinky finger, the ring finger, middle finger, index finger, and thumb. And you see in the sensory cortex, this is all kind of well behaved. Neighboring fingers on the hand are neighboring in the brain. And we have this kind of homunculus model. And it's pretty gigantic. Like the huge chunk of the sensory cortex is responsible for these finger representations. And you don't really need extremely high resolutions for that. Now, the resolution comes in when you look at it in the other side, namely the motor cortex, where we have this little cave over here, the hand knob. And again, I'm projecting the individual fingers, the pinky finger. Now we have 2 ring fingers, and then we have 2 middle fingers, 2 index fingers, and 2 thumbs. And they're very, very close together. So the high resolution information here reveals things that we could not see at lower resolutions, namely that there are multiple finger representations that at conventional resolutions would just be lumped together, which might be better appreciable also when you look at a kind of slice like that, where on the bottom left, you have the gigantic sensory homunculus somatotopic representations, and in the motor cortex, we have then these very cramped together small finger representations, where the entire finger representation is basically as big as the cortical thickness. And this was kind of surprising to us. And this is new information that we only saw with high resolution, not with lower resolution. And we've found that this is actually very well consistent across different participants, where we always have these multiple patches that are also correspondingly nicely with different movement actions, like if you're grasping something or retraction movements. So the motor cortex at low resolution is a nice homunculus where each spine is represented next to each other, but within the spines, in the mesoscopic regime of these high resolutions, you find that there's actually more information that we can harvest there. Namely, see that the motor cortex is more an action map than a body part representation. I don't see my time. Maybe I move this. Okay, I have time. So all these data and examples that I showed you so far are all being conducted at 7 Tesla scanners that Tyler introduced. And there are about 150 scanners out there. So until now, still the high resolution and the high field community are kind of really highly overlapping. And it's kind of a rich man club. There are these few prestigious research institutions that have 7 Tesla scanners. And the number of 3 Tesla scanners is about 2 orders of magnitude higher. And 3 Tesla is already pretty rare compared to clinical scanners of 1.5 scanners and lower. So I want to spend a few slides now to kind of describe this interaction of ultra high field imaging, 7 Tesla, and high resolution imaging, namely by using these 2 graphs that I think you will see several times during the summer course. And actually this graph is something that I grabbed from Peter Banettini's lecture last week. So this is showing the relationship, the nonlinear relationship between signal quality, the SNR that Tyler already introduced to some degree, with respect to signal stability, what we care more about in functional imaging. And the higher the field strength you have, the more sensitivity, overall signal quality, SNR, and also the lower the resolutions, the more magnetization you have, the higher the image quality. However, there's this weird nonlinear relationship that at some point, having higher image quality doesn't really buy you a lot with respect to the stability. And this is something that Renai already mentioned on Tuesday, that in this domain, you're really limited by the scanner noise, thermal noise. And at some point, you're not really limited by the scanner noise anymore. You're more limited by the brain noise, because the brain is always kind of doing stuff, pulsating, there's respiration and so on. At the same time, we also have a nonlinear relationship between stability and minimum time that you need to scan for a detection threshold of a given effect size. And I would like to give you a more intuitive understanding of those graphs and what this means for high resolution imaging with 2 experiments that I was conducting a while ago. Namely, here in this experiment, I was interested in finger tapping, and specifically on the ipsilateral side, there's this inhibition, a reduction specifically in the sensory cortex, which is a tiny effect size, 0.5% signal decrease. And even though we had big voxels, the effect was small, so we had to scan a lot. We have to scan 30 minutes to really see this effect robustly. Now, on the right-hand side, I'm looking at the same task, but the contralateral side, which has a huge effect size, 5%. I was looking at it at very high resolutions though, so my noise level was not the best, and I had to scan about 30 minutes to get it robust across people. So in both cases, I had about 30 minutes. that needed data to see something. And in both cases, I was really sensitivity limited. So what would you expect? Are both cases similarly benefiting from going to higher field strength? The answer is no, right? So if we look at those graphs for 7 Tesla, in the red case here, we are starting off with big voxels, so we have pretty good SNR to begin with. So going from 3 Tesla to 7 Tesla improves our image quality by a factor 3, but still our stability does not really improve a lot because we are more limited by the brain noise, not the scanner noise. And this means that even though we have pretty good sensitivity, because of our small effect size, with this increase in stability that's pretty small, we even have a smaller benefit in minimum scan time needed. So we maybe increase the necessary scan time from 30 minutes to 28 minutes or numbers like that. Now for the other case, the blue-grayish case, we are starting off with very low stability to begin with. This means that tripling the sensitivity and image quality actually triples the stability. And because we have very big effect sizes, we are in this nonlinear regime where a tiny improvements in sensitivity really substantially reduces the necessary scan time needed. So here we could actually go from 30 minutes, more than a factor of 3, to 6 minutes scan time to see something if we would go from 3 Tesla to 7 Tesla. At the end of the day, you can do high resolution on any scanner you want. You have different challenges. And for the same person in 3 Tesla, 7 Tesla, 9.4 Tesla, you have different challenges, but the best scanner at the end is always the scanner you have access to. At 3 Tesla, you just need to scan very, very long, but it's possible. At 9.4 Tesla scanners, you don't need to scan as long, but you need to invest hours of prep work. You need to have a team of engineers helping you with these kind of prototype scanners. So you might end up with more time investment after all. At 7 Tesla, you have a nice sweet spot there, which kind of is very efficient to get you from A to B. 3 Tesla is the kind of low-cost version, and 9.4 Tesla, you can break records, yes, but it's challenging to get there. As Tyler kind of introduced in her kind of two-dimensional space of scan time and what was it, image quality. This is basically a different spin on the same kind of compromises that we can spend our time at the scanner in the encoding limited regime to get a lot of voxels and that are very, very small, or we spend it to get a lot of voxels to cover a large area. But we cannot really do both at the same time, right? We have either resolution or coverage, or you can focus on a few voxels and scan them and sample them very, very often. So as soon as you want to push one of those parameters, you need to make sacrifices with the other ones. And I would like to give you a few ideas on where the field stands with respect to pushing either one of those, starting with coverage. So I showed you this metaphor of the truck engine, that when we look at a part of the brain, with low resolutions, it's just like understanding the brain, like looking at a truck engine with infrared camera. Now with the high resolutions that we get, we actually have an idea of kind of where signal comes in and where information flows out, and namely looking at these different little stripes of the motor cortex. The cheeky thing of me was that I introduced this as a regular truck engine that's embedded in a system. That's not really the case in the motor cortex. Namely, the motor cortex is not like a regular truck engine. It's like the biggest engine there is. It's isolated, and it's like 4 millimeters thick. Any other cortical areas are in the range of maybe 2 millimeters or 3 millimeters max. And it's relatively easy in this isolated case to look at input versus output or whatever. Now, for layer fMRI to really work across areas, we need to look at it also for regular sized cortices and how they're kind of embedded in a given system. Namely, it's not enough to just cover the motor cortex. We need to see how the motor cortex is embedded in the sensory motor system and how the sensory motor system is embedded in the rest of the brain. So there was a lot of efforts in the last decades to increase the resolution and coverage at the same time. And one of the studies that I wanted to highlight here is an open data set that you can download, which is only individual brains, but those brains are scanned 52 times with the 15 minutes movie experiment. And you can kind of quantify kind of the issues and the stability and repeatability. And to get the whole brain takes about 5 seconds. And if you want to have more than one contrast that I will talk about end of the month, you even have longer temporal sampling than, like slower temporal sampling than the time it takes to acquire one volume. If you don't care much about coverage, but more care about temporal resolution, you can also go there and only cover a tiny patch of the cortex, but then those 16 slices capture them very, very rapidly, for example, to look at these temporal differences, for example, across different cortical depths where input is coming in, and then certain layers have an earlier peak time compared to others. Last, let's look at the resolution, how much the field can push it there. And I think some of the highest resolution images in humans are the ones acquired with a very specific head gradient, a scanner that's not kind of originally built for clinical imaging, like our whole body scanners, but rather a kind of a head scanner with an optimized gradient. And there you can push to voxel volumes that are smaller than 0.1 microliter. And namely, even in these very thin cortices, see indications of the signal distributions within the cortex. At NIH, we don't have scanners like that available, unfortunately, or not yet for that matter. But still, we can kind of try to make up for the limited encoding power of this hardware by basically spreading it across time, basically sampling the same case space, but then because we cannot do it so fast, do it across different repetitions and then fill that in retrospectively to also get to similar resolutions here and see these very fine scale details that I've introduced earlier. So I think resolution is really something that you can push even beyond the resolutions that you usually need. And namely, the standard is about 0.8 millimeter for depth-dependent fMRI. And to give you an idea of the entire spectrum of resolutions, I would like to show you this slide here where Saskia Bollmann summarized with a meta-analysis of fMRI that the median resolution of most fMRI is bigger than 3 millimeters. And that's like not very coarse. It's the kind of the size of 2 quarter coins on top of each other. And it's kind of the standard for regular fMRI. Now for layer fMRI studies that are aiming to look at cortical depth, the standard has emerged to be in the range of 0.8 millimeter resolutions. It's the thickness of a credit card, quite thin. And because each side of a cube is getting smaller, this means that the volume is reduced by a cubic law. This is already like a huge reduction in volume, more than an order of magnitude. Now, the highest resolutions, some of the highest resolutions in humans are going to 0.4 with these dedicated hardwares, where you can see it's almost an additional order of magnitude. So you have a lot of degree of freedom to play with the resolutions. And even though this is kind of the limit of what human fMRI is doing right now, we can still dream and think about the things we need to work on in the next 10 years to get even higher. And we are inspired by results like this. Like in the animal literature, people are usually ahead with respect to resolution by a few decades or so. They are going down to 200 micron resolution. Now, the thickness of a banknote, that's very, very, very small. It's all another half order of magnitude over here. And if in animals they can do it, we will be able to do this as well, right? The laws of physics and understanding the noise, they don't care about which species you're in, right? You have certain different constraints, but I'm certain that at some point we will be able to optimize our resolutions to get there too, where you have not kind of 2 stripes across cortical depth, but see all these kind of wiggles in there. And to dream even further about the ultimate resolutions in fMRI, we can also be inspired by structural imaging. So some of the highest structural results out there are looking at 12 micrometer. And this is now the size of these little fibers in the banknote. And of course, in ex vivo imaging, you have more degrees of freedom, right? You can scan for an entire weekend. But a weekend is not that long. Like it's not something that we can overcome. A weekend is like 50 hours. 50 hours just 7 squared. So we should be a factor 7 away from what they can do in ex vivo imaging. Why are we 5 orders of magnitude away? So the physical limits are still kind of, there's a lot of degree of freedom for people like us to work on to get to higher and higher resolutions. So with all these resolutions that are available, what is holding us back at the moment? I think right now, most of the limits at conventional fMRI, and the reason why people are not pushing to higher resolution than 0.8 is because raw data look like that, which is kind of good raw data, and you can get a lot of neuroscience questions answered with those kind of data. But if you look carefully, there are all kind of weird shit happening. There are these kind of low spatial frequency ghosting, there are these interference patterns, and those are kind of the best data they are. They are taken from the top 10 labs around the world. And so this is the kind of things that we are currently working on. And it's hard to work on those because it's kind of much easier in the current funding or job environment to find collaborators who want to get the best data and that are currently available to answer their brain questions, it's much harder to get positions or funding or grants for questions like to not get the best data, but to worry about the issues that we have at the moment to get the best methods that then in 10 years or 5 years will get us the best data. So to put all this into perspective of different research fields here at NIH, I would like to kind of show this spectrum as the last slide of this presentation, that kind of we are all dependent on each other. And we have heard a few talks already at the early days about how Peter was wiring his own gradients, then to do the experiments in the same day in the afternoon. He grabbed the data overnight to do his analysis. And on the next day, he was able to kind of look at how he needs to rewire the gradient, right? So in the early days, there was a very efficient feedback loop, and each person doing fMRI was kind of really on top of each different space of performing an fMRI experiment. And unfortunately, or fortunately, fMRI has really evolved since then, which is awesome because hardware got better, everything got better and more specialized, and each different step in an fMRI experiment has its entire journals, their entire conferences in their individual disciplines. However, it means that the crosstalk is not as big, and maybe also the appreciation is for each other is not as high anymore. Maybe with the example that there are these new gradients that are being developed from engineers, and they're putting a lot of mental energy in it. And then there are people like sequence developers, and maybe when I mentioned this to some degree, that saying, yeah, you have these nice switching rates, they're cool and nice, but at the end, it's useless. Like, I need to get it to images, right? This needs to be safe. It needs to work in a human, right? So then people like when I come along and turn this into a beautiful image, right? And this is an individual discipline of science, right? To convert hardware to images. And then I can come along and say, well, this is a nice image, but I care more about than just images. It's pretty pictures, but I care about stability. I want to convert this into an activation map. And then I come along and think about it hard and convert this to an activation map, right? And then people in the maybe third floor come along and say, well, this doesn't have substance, right? This is just a pretty picture. I need more than activation maps. I need to learn how this tells me something about the brain is wired, and if I can test my theories about cognition with respect to the wiring patterns of feed forward or feedback or something. Which again is kind of then just serving the next disciplines, right? If you do these kind of, if you have your circuit understood, then the next people come along and say, well, you have your circuit now well understood for your nice task, but at the end, I need to relate to behavior, to symptoms, right? We are here at the NIH. And then even when they figure it out in their big studies, the medical doctor says, well, this is sweet, but it's not helpful for me because now you know how this little layer is affecting my Parkinson's patients. But at my next patient at 3 o'clock, I cannot tell them to just activate his layer 3 neurons more. So what I want to say with this very long slide is that each individual spot along this hierarchy that we are aiming for at NIH has its justification for existence. And so far, I was really focusing on the early part here. And in the remainder of the summer course lecture, we have a lot of stuff and a lot of expertise at NIH that is focusing on the right part of the graph. And with this, I want to thank all of you and my colleagues for their help. Are there any questions? It is 14:59. So we are at full hour. Thank you very much.